Oh, good morning, everybody. I think you guys caught me catching a sip of coffee. Good morning, everybody, and welcome to the Keen 1, or Keen of New England meeting. So I'm here with a group of students from UMass Boston. We'll have people here as well from other universities as well. Today, we're going to be talking about the methods of the Kelp Ecosystem Ecology Network. And so much like yesterday in our data hangout, for those of you who are watching online, I'll be screen sharing. Since I'll be largely talking to people here, you will see what I'm showing everybody here. So without further ado, let's get started. There will be some time for introductions in a minute, and we'll go over that as well. Pull up our slideshow for today and maximize this for everybody. All right, so good morning, everybody, and welcome to the Kelp Ecosystem Ecology Network meeting. And this is in particular the Keen of New England, or Keen 1. So today what we're going to be doing is going over the materials and methods for Keen of New England, although what we're going to talk about today is broader and more broadly applicable to all of the Keen nodes. So for those of you who are in the room, we are webcasting this to an international audience. So that should be useful, as a number of people wanted to see exactly what we're doing here, so they could import that to their own site, since we are all indeed using the same research methods. And I'll be talking a lot about standardization as we go out throughout today. So I just wanted to start off with a thank you. This is by far from incomplete, but this is just some of the people who are responsible in developing the materials and methods that we talk about with today. So this is just a few people, and there are many, many others. The materials and methods that we're going to be talking about today for a Kelp Ecosystem Ecology Network sites have been rigorously gone through by a number of people. They're adapted from different programs, and so a lot of people are responsible for what you're talking about today. If you have any questions, comments, or issues with it, though, I'm the one at fault, so bring it up with me. They've been great. The purpose of today, as well as tomorrow's meeting, is to walk out of here and get up and running. We have this meeting every year because every year there are new people. Every year everybody's brains erode a little bit and we forget what we're doing exactly. So the purpose of today's methods day and tomorrow's species identification day is so that when we hit the water in the coming weeks, our brains are fresh and we are ready to get out there and sample and collect high quality data. If this is your first meeting, welcome. If this is not your first network meeting, I'm glad you're here again. I'm sure your brain has eroded, not as much as mine, probably much less, but I find this refresher really useful every year. I want to start off with, if you know of other labs or other people who are interested in the work we're doing, this is an open network. So the Kelp Ecosystem Ecology Network, which I'll be talking about its network goals in just a minute, is an open collaborative network focused on kelp ecosystems around the world. If you know some people who are interested in this project, either they are have a lab themselves and are interested in setting up a monitoring site, or they're just divers who are, they're scientific divers who are interested in working with labs that have ongoing sites, please let me know. We're open, we're always looking for people to join. You can check out our website at kelpecosystems.org. You can go and talk to me and subscribe to one of our Google groups. Right now we have a Kelp Ecosystems Google groups, which is the overall Keen network group. We also post lots of other things there. People send papers through, they send job opportunities, they send grad student and postdoc openings. So it's a useful listserv to be on. We also have a local Keen 1 listserv. So this is the Keen 1 Google group, particularly if you're a scientific diver in New England who's interested in knowing what's going on with Keen 1. Let me know, we'll get you signed up for this. For those of you who are online and are listening from the West Coast, anywhere from the Bering to Baja, we've also recently set up a Keen B2B listserv. And so Matt Edwards, Allison Haupt, and I will be coordinating that soon, and there will be more discussion on that listserv. So that started up. If you want to get on that listserv, that will be there. And hopefully others will be coming online shortly. Overall, if at the end of this presentation or at any time you have questions about Keen, I am the network coordinator, so please contact me. And and I'll give my contact information out later, or I'm easy to find, Jarrett Burns, burnslab.net. So what are we going to do today? Here's a rough agenda for today. It may be plus or minus a little bit. And for those of you who are online, this is the first time I've done a Google Hangout with multiple sessions, so there will be a little bit of tech troubleshooting of making sure you're still online. I don't know if these Hangouts actually stop and start or not. So we're going to begin with a welcome and talk about what is Keen. And then we're also going to have introductions, both of people who are in the room as well as anyone who's online, so we can see who's tuning in. For those of you who are watching this and are in the future and aren't watching live online, if you could just add an introduction of yourself in the comments of this video on YouTube, that would be great. We're then going to go through a review of all of the protocols. So I've scheduled this roughly for 10 to 11. It may go a little longer, it may go a little shorter. We're going to talk about each of the protocols. We'll first begin by talking about the protocol for a removal experiment. That's a little bit in flux, as we'll talk about, and hopefully we can have some open discussion about ideas or thoughts about changing that experiment a little bit. I'll present the way we're doing it now and the way that a few of us within the network have been talking about changing it for the future. We'll take a break, because you all need a break from me, definitely. And then when we come back, we'll talk about data collection. So we'll go through some of the data sheets and how to use them. And we will talk a bit about data entry, which seems like a tedious thing to talk about. But I want to go over the various forms and things that we have to introduce you to them. The reason we're doing that is I've spent the last month or so, at least, coordinating all of the data that's been sent to us from different Keen 1 network members. And so I've made some changes to that process to make it basically harder to do something wrong. Spent a lot of time cleaning a lot of data that was a little, you know, in general, great. And I'll admit 90% of the errors came from my lab. Everybody else was great. And so I want to kind of go through those forms a little bit. At noon, we'll have a who is doing what where conversation. So for the PIs who are online, we're going to talk about who's doing what, where, when, and if they need help. At 12:30, we're going to break for lunch, and then we'll come back in the afternoon. So for those of you who are tuning in online, basically after the data collection and entry and who's doing what where conversation, we'll shut this off. In the afternoon, we're going to do a live practice. And I would suggest for those of you who are watching online, if your lab is going to do this, there are 2 steps of practice that I would recommend that you do before you actually go out and start taking live data. And I'll talk about both of those as we go through today. So I'm going to begin with an overview of Keen. For some of you, this is old hat. For some of you, this is quite new, because this is your first time here. As a research network, our goal is to understand the impact of global change on the world's kelp forests. And I don't just mean that in terms of kelps, I mean that in terms of kelps and all of the species associated with them. To some extent, you can think of us as a biodiversity observation network. So we are trying to make some contributions to the bond world as well. But we really want to understand long-term change in the world's kelp forests. And in particular, we're interested in thinking about long-term change because of temperature change. So this is one of the maps from the Intergovernmental Panel on Climate Change Working Group 5 report, the physical basis. This is just degrees Celsius change from the 1901 to 2012. And we know that much of the world's oceans are warming. There's, of course, a great degree of variability in how much warming is occurring. But a lot of the world's oceans are warming. Kelps, which we are all interested in, which we are all fascinated by and passionate about. And you're in the Kelp Ecosystem Ecology Network, so I'm going to assume that you're like totally gaga over kelp. And if you're not, what's wrong with you? Kelps are cold water, temperate algal species, and so they should respond to temperature change. We have seen evidence of this in multiple places around the world. There's some really crazy rain shifts going on right now in Europe, Japan, various other places. There's some exceptions that prove the rule, like in South Africa. We know that there's a temperature component to change, but to what extent the long-term creeping change of climate change is actually affecting kelps, that's something that we're really only starting to quantify on a global scale. Is climate change generally affecting kelp forests everywhere? We think it should. We don't know necessarily if there's a common set of mechanisms. But we have some suspicions, and that's why we have this network. Keen 1, I feel like, is a particularly important set of data points within this study because New England is in the hot seat. So here is a figure from the Pershing et al. 2015 paper looking at temperature change and its effects on cod over time. This was published in 2015. And one of the things this analysis showed is in looking at the distribution of temperature change in the world's oceans, the Gulf of Maine is in like the tail of temperature change. We're one of the fastest warming places on Earth. Not only that, but the Gulf of Maine has a tremendous, basically, temperature climb. We go from, not just the Gulf of Maine, but New England as a whole, we go from quite warm Gulf Stream influenced waters down in, say, Long Island, the southern distribution limit of kelps in New England, up to quite cold waters as you move around the corner and up into Canada. So we've got a really big thermal gradient, so if you want to think of like space for time substitution, but we're also changing at an incredibly rapid rate. So it's an ideal place. So Keen 1 members, we are like perfectly positioned to understand this stuff. The network has a few specific aims. So I gave you our sort of general network goal, but specifically the network as a whole is set out to do a few things, and we're just starting to begin to bring together a number of data sets to address these. The first is unifying and finding past kelp forest ecology data sets for synthesis of climate influences. I'll talk a bit about that. The second is a climate change kelp forest experiment or experiments along thermal gradients. The third is global standardized kelp forest monitoring along these temperature gradients. So again, to try and look across space and just monitor things over time. And last is start to unify and synchronize data sets within the kelper community. So there are a number of long-term kelp forest monitoring programs out there already. Some great ones in Santa Barbara, those run by PISCO, up in Haida and others. And one of the next steps we're going to be taking is trying to take these programs that they've got their own methods and start to link them up to ours and see how do they compare. So I'm going to pause for a minute and just talk about who are we. I want to know who's in this room, what your interests are in particular, and just sort of get a sense of who is here. So let me tab out of that. OK. Stop screen sharing. Screen sharing been on this whole time? OK, great. I'm just worried that I came over here and it was looking at a picture of me, and hopefully it was not off. So we'll find that out later. And if it was off, well, sorry about that. OK, so we've got a live Google chat. We'll see if anybody's there. Give that a few seconds to see if anybody's participating live. Please say hello in the Google chat. And in the meantime, who are you? Who's in this room? Let's just go around. Not everybody knows each other, so let's do a quick round of introductions. You all know me, first off. So I'm Jared Burns. I'm here at UMass Boston. I coordinate Keen 1, and many of you are in my lab and actually will be doing work with us. So who else is here today?
Hello, I'm Jeremiah. I just started 3 days ago, coming from California, and I'll be working on Ted's project with wave sensors.
Okay, sweet.
Helping out with Keen.
And Kelp.
Okay.
Yes.
Oh, I'm Ted Lyman. I work for Jarrett in the Burns lab and love kelp and I love waves. And so we have a project of doing wave sensors in kelp.
I'm Katie. I met Jarrett and Ted at the Boston Sea Rovers dive show in March, and I'm pretty excited about helping them out because I have a lot of free time.
Katie did the underwater research methods class this summer in Denver.
Okay.
Hi, I'm Felicia. I work in Jared's lab. I'm also part of an REU program right now, which is research experience for undergrads. I'm under Jared's wing, and I am working on kelp abundance and predation rate.
I'm Becky Mahallam. I'm a PhD student in Jared's lab, and I'm interested in how subtidal species are responding to the rapidly warming Gulf of Maine. And I will probably be hounding you all to dive into the Keen stuff a lot.
Yeah, so Brecky's the grad student who's sort of in charge of coordinating Keen within my lab. So She is your go-to for all things keen within the Burns Lab this summer.
She's keen for keen.
She's very keen for keen.
I'm Nelson. I work in the Burns Lab, and I'm kind of the gopher. So if you need anything, just let me know. See what I can do, or I can find somebody who can.
So I'm Rob Holberg. I'm a PhD student in the Hannigan Lab in the School of the Environment here at UMass Boston. I'm studying the effects of ocean acidification on otolith development to reef fishes. And I'm also deeply involved in projects looking to combat wildlife crime, saltwater crime.
Cool. Yeah, if you guys have a CFET and ever want to put it out on our sites, we're game. Yeah.
I'm Isaac Rosenthal. I'm in the Burns Lab too. I studied kelp mostly with remote sensing data. I'm interested in how kelp populations function over the last 30 years across most of the world. And Keen is a great institution.
Awesome. Do you have anybody online, Isaac? Cricket. Oh, hey. Jeff, why don't you introduce yourself? Or not. I guess we're a little scary. Yeah, there is. Well, if you are following along online, please feel free to introduce yourself as we go forward. I'd love to hear who is online and watching. And as I mentioned before, if you're not able to participate in the live chat, please hop on and say who you are in the comments. Oh, okay, Jeff is the DSO down at University of Connecticut. Cool, Jeff. We should talk later. I know I've been communicating with a couple of you guys about setting out a keen site at the southern range limit of kelps here in New England. So we'll chat later, perhaps around lunchtime. Okay, so I'm really glad that you're all here today, as well as others who will be joining in, and we will continue from there. So screen sharing working. Yeah? Oh, good. Froze up something. It is. Okay, so for those of you who are interested in the Kelp Ecosystem Ecology Network, again, kelpecosystems.org is our website. I will not only be linking this presentation there, both the slides as well as the video, but all of our materials, methods, and data are available there. So if after all of this you're super excited and want to learn more, just go to the Keen One website, or the Keen website. Keen One doesn't have its own site, just our own Google group. One thing about Keen, again, this is both for those of you in the room who end up moving on to other places, as well as those online. Keen does have different regional coordinators. So here in the northeast of the US and Canada, I'm the coordinator for the Gulf of Maine New England Network, Keen 1. Pat Gagnon is the coordinator for Canada. So Pat and I have been talking about how to get things going up there. But we have a variety of different other regions around the world, and each region has its coordinator. So Alison Haupt, who did her work here previously, is working actually with Matt Edwards to coordinate the Baja to Bering region, along with Brenda Konar. There are a variety of other coordinators around the world. If you're interested in getting a site involved in Keen, contact me. I'll hook you up with your regional coordinator, and they'll help you coordinate what you're doing with the central data that they're collecting before it gets passed on to me. All right, so for those of you who aren't familiar with research networks, I wanted to give a little bit of background on research networks, what they are, why we're doing what we're doing. This is sort of my, I guess, philosophical indoctrination portion of the morning. So Keen really started with data synthesis. A number of us participated in a working group at the National Center for Ecological Analysis and Synthesis in 2012 and 2013. And one of our goals there was to understand what are long-term trends in kelp forests. The first thing we did, really the major thing we've published so far, was going in and just trying to find all of the time series of kelp forests that exist ever in the world that we could. And we put together a data set of roughly a little over 8,000 observations coming from about 1,400 different sites around the world. Shown here, we've got the distribution of those sites. So warm colors are places where we have a lot of different time series, where time series is defined very, very generously as anything with at least 3 data points. You can see there are certain portions of the world in gray where kelp is found, but we don't actually have any time series data for those regions of the world. So there is a lot of the world where we don't have kelp time series. It's just not there. We don't know what's going on. You can also see a lot of regions of the world that are in cooler colors, that we do not have as much data that we'd like. So there are many regions of the world that we want more data. At this working group, we put together a number of synthetic efforts, both this analysis of all kelp time series we could find. We've also started a citizen science project that Isaac and others are working on, looking at satellite mapping of canopy kelps around the world for a 30-year data set from the Landsat satellite, as well as a variety of other different projects, all of which are making their way towards fruition. The first one, for anyone who's interested, is this paper by Kira Krumhantel, as well as a variety of network members, looking at global patterns of kelp forest change over the past half century. This was the data set where we really brought together all of our different time series. So you can see here is all of the world's kelp time series all in one place. It's a giant kind of mess, but what we really found within this analysis is that in order to understand change in kelp forests, there's a lot of variability and change from region to region. In some places there are increases, some places there are decreases, some places it's staying the same. You know, if, for example, we just look at New England, so these are the data sets from essentially the region that Keene 1 covers, as well as out of Nova Scotia. And shown here, these solid colored lines are the lines of best fit from a Bayesian hierarchical model, with that gray area as the credible interval around that fit. We kind of see a trend towards long-term declines, although actually even in the final analysis, there's more uncertainty than that. We find in general within this province that there's a high degree of variability. One of the main messages that came out of this synthetic working group is that while we do have a lot of data out there, there's such variability in methods and such variability in place and such a lack of systematic effort in terms of trying to link these data sets with climate drivers, that it's very, very difficult for us to actually derive meaningful data about long-term change in kelp forests and link it to things like climate change or other global change drivers. And so at the end of the meeting, we really came together and decided that something like Keene needed to exist so that we did have large-scale standardized databases so that we could really look across a region and ask coordinated questions. I'll actually give a great example, one that I really like. So Bob Stanek, who's up at the University of Maine and is a member of Keene 1, has been serving in a really great advisory capacity and kicking our butts in terms of asking questions the right way. He and I have been discussing a lot about even the New England data here and the tremendous amount of variability, and thinking a lot about, well, is there variability across space? Even within the Gulf of Maine, are there places that are increases and places that are decreasing, and are there reasons why that if we just had long-term data, we could begin to understand that? And so people like Doug Rasher and Theo Suskowitz are starting to try and think about that question a little bit more of a coordinated way as we put together all of this Keene 1 data and begin to think spatially and begin to think about what are the drivers of variability in kelp forests. So this is why Keene 1 needs to exist. We need to make sure that we are doing something similar so that our data is really broadly comparable. This is a great start, but it's not necessarily going to get the definitive answer unless we all are doing things the same way. If you are interested in this data, though, from the Krumhantel et al. Paper, I talked about this a lot yesterday, but the kelp time database, as we've been calling it, is available from a wonderful data repository for temperate reef data called Temperate ReefBase. If you are a temperate reefer, this is where you should be putting your data for long-term archiving and access. This is a project run by the University of Tasmania, Craig Johnson and his colleagues. Just go to temperatrereefbase.imas.edu.au, and you'll be able to go in and search down this data. And this is by no means the only data set there. There are a ton of different incredible data sets worth taking a look at to understand all sorts of different patterns in our temperate oceans. As I mentioned before, we also have a citizen science project. It's about to sort of be relaunched, but if you want to take a look at where it's gone so far, check out floatingforest.org. This is a project that right now is funded by NASA. It's a collaboration between UMass Boston, UCLA, and the Adler Planetarium as part of KEEN. And here we're trying to look at satellite data to build a 30-year global database of kelp abundance for you. Some of that data from the sort of pre-citizen science, so this is verified data, is available from the Santa Barbara Coastal LTER's website. And as our data is made accessible, which is one of Isaac's projects this summer, that data will also be available on Temperate ReefBase. And so this is kind of what it looks like. You go to the site, you see a kelp forest, you circle it, and boom, that's data. That is one slice of time and one place. And as we build that up, we'll be able to understand, hopefully, the global changes in canopy kelp distributions. And it works. So far, we find there is actually a good deal of consensus between users, and we can build really nice consensus maps. Underlying all of this work is a conceptual model. So at the NCEAS meeting, we talked about a lot of different ways that temperature change in particular could alter kelp forests. And the model we really came up with was trying to think about hysteresis in kelp forests. That many of these forests do just fine as you warm the waters until you hit a certain tipping point. So, for example, if you look at the Western Australia, you see in some of Thomas Wernberg's work that adult kelps are fine with temperature change until you hit a certain major warming event. And that's really what affects kelps in the end. We see potentially similar trends here in New England. For example, if you look back at some of Val Gerard's wonderful work in Long Island when she was at Stony Brook, southern kelps are super tolerant of warm waters, again, until you hit a tipping point. And so this is sort of the underlying model we have of how kelps might change over time. And really the big idea that's arisen out of many, many different kelp systems and things that we've learned, particularly from looking at formation of urchin barrens, is that kelps will typically hang around even in the face of slow creeping stresses like temperature change until some acute stress hits them, so that there's some disturbance that triggers and kills off adult kelps. And after that disturbance event, if conditions are not good, you will not see recovery. If conditions are cool enough, kelps will recover. I mean, kelps really are the badasses of recovery in marine ecosystems. They're just amazing in terms of their abilities to bounce back. However, the thing about climate change is that once you pass a threshold, we find particularly things like kelp gametophytes are less able to hack it in the face of warmer waters in some systems, for example. And so when you remove the adults, there's less of a gametophyte bank left to enable recovery. So it's really this idea of disturbance crossed by temperature leads to less recovery, and that there's some point where after you, even if you have what looks like a healthy adult forest, after some tipping point of temperatures, that disturbance event will lead to some problems and a lack of recovery of that forest. So this is sort of the underlying conceptual model. And the question that we have is keen to answer is this general? And is this true for the community as well? So I talked about that in terms of kelps. We all know that kelps are foundation species and facilitate a wide diversity of different species. So if we were to instead put community structure on that y-axis instead of just kelp abundance, do we see the same thing? Do we see the same precipitous state shift? Or do things that come in and say replace kelps allow communities to stay roughly the same? Or is there a direct effect of temperature on those communities? These are all things that we would like to tease apart. What are the direct versus indirect effects of climate change on kelp forests, et cetera? In order to answer that, we need big standardized data sets of biodiversity in kelp forests. So who knows? While our synthesis was really good, there were some problems. The studies are not designed for this model that I just showed to you. There is a huge amount of variation in methodology, such that trying to look at a model like that is not possible across the data that we assembled. In some areas, there's sort of a lack of proper regional variation and climatic variability. So we had like one data set from one region. So you can't necessarily look at variation across that region, which is necessary. And then sometimes there's excessive variation in other things that we weren't able to really control for in a meaningful way. The answer is to go back and look at this map and ask, what if each of these sites had one experimental plot? What if each of these regions had a network of observations? And this is something that's become relatively common in ecology, this idea of sort of bootstrapping up a network from a variety of scientists like all of you, and in areas replicating the same experiments and same observational data sets and then deriving inferences from these. I think NutNet is sort of one of the really great examples of this. This is the nutrient network that was founded by a number of scientists to look at the variability in responses of grassland communities to fertilization treatments. So Liz Borer, Eric Seablum, and many, many, many others who run this network. And this has been an incredibly powerful network with all of the observational and experimental sites that they have spread across the globe, shown at the bottom. They conduct standardized monitoring as well as standardized manipulations. And from this has sprung a wealth of incredibly valuable analyses addressing everything from very applied questions to sort of very broad theoretical questions in ecology that to this date we haven't been able to answer nearly as well without an experimental network like this. In marine realms, there's the Zoster Experimental Network, which I know that some people within Keene have been involved in. This is a network set up by Emmett Duffy, again consisting of some observational data as well as manipulations of amphipods and nutrients. So amphipod manipulations are done through carbaryl or an insecticide that excludes amphipods without the need of a cage. And this ZEN network has been able to discover broad patterns across the planet in terms of how seagrasses respond to top-down and bottom-up control, answering some of the big questions within seagrass ecology that heretofore have been trickier to deal with in such a coordinated way. He also uses fancy modeling techniques that I happen to like a lot. But that's my personal modeling bias, I'll admit that. So here really what we're trying to do is create both an experimental and observational network to develop clear scientific goals and questions, to implement identical treatments and sampling designs, something that's simple, relatively inexpensive, although I acknowledge we are doing subtitle work, which is never as cheap as you want it to be, and using something that's flexible so that people can implement it with some additional room to do their own work that uses our base design and builds off of it. We have started with a critical mass. So Keene 1 is really that sort of initial nugget where we've started to build a data set over the past couple of years, and we're spreading this around the world. We have fairly clear ground rules for participation. Just do one thing, an experiment or an observation. That's it. If you do that, you're in, you have access to the data. Welcome. And the benefits are data, co-authorships, collaborative networks, and hopefully some really cool analyses coming up that we'll all get cited on. So woo, citation career building. And lastly, we've begun to implement a plan for data management. So now we actually have the data factory up and running. We have not only a really nice quality control process that's fairly automated, but we now also have a data host for long-term data storage and management. So that pipeline is now present. And just to, again, like evangelize a little bit, when we think about the effort versus reward within science, if we think about individual effort and the reward that we all get as scientists in terms of generality, data, publications, and collaborative networks that, for example, you early career folk in this room might want to build as you go on in your careers. You know, if you're one person working at one site, you can put in the effort you can put in, you get some reward. Maybe it's big, maybe it's small, it's maybe not huge. If you, for example, sample a whole region, you may be able to get much more information out of that, more generality, higher reward, but man, that is a lot of individual effort. If you were to try and do something global, that's a lot of effort. It's a high degree of effort, but it's also a high degree of reward. And there are some people who are really, really good at this and have built their careers on it. And my hat's off to them for the boundless amounts of energy that they have. Some of whom we're fortunate enough to have in this network and are really great at sort of kicking our asses in terms of how this stuff needs to work. The model of something like Keene, ZEN, or NutNet is that each individual scientist puts in effort at one site. So low effort, relatively, as much as it ever can be. But given that there are many hands together, many hands make light work. And the types of analyses and data that can come out of this will be beneficial to everybody. So many hands, light work, high reward. So what does it mean to be part of KEEN? For PIs and labs, the idea is to implement just one protocol, either the survey or experimental protocol at one site. And if you do that, you're good. You're in, you have full access to everything from the get-go as well as the network. Second, share data within the network. So don't just take the data and do it and then walk away with it, but really this is about data sharing. You will receive credit for data contributed. So once we, for example, put the data from KEEN 1 thus far onto TRB, each individual lab has a citation. So if someone uses your data in the future, they will cite it. You will receive credit for that. Receiving co-authorship on products that use your data. So there's a policy for that as well as access to network of collaborators for future projects. So again, for technicians who are working with this in network, this is maybe where things pop up a little bit more. By being part of this project, any lab around the world knows that, oh, you've worked on this, you know what you're doing, you've been involved in this effort. And so if we're doing something similar, it's going to be much easier for you to match in on that. There have already been some great grad student opportunities that have been shared within KEEN that I and others have sent around to our students. So a lot of opportunities here. You know, heck, Jeremiah, I hired you this summer because I sent out an email to KEEN 1 being like, hey, I need a tech. And Nicole wrote back, oh yeah, I have this great guy, you should work for you. You know, Austin sent back a number of wonderful people as well. So we had this like pool of people that came directly through the network. So nice little networking benefit here for those of you who are at that stage within your careers. So again, one site of one protocol per team equals a lot of sites if many people participate. We get common data. It's data we can all work with. We don't have to reinvent the wheel. And we are a biodiversity observation network, de facto. Bonds are becoming more prevalent. For those of you that are aware of the Intergovernmental Panel on Biodiversity and Ecosystem Services, or IPBES, one of their efforts is the bond in a box. And there's a real push to try and expand this idea of biodiversity observation networks. We know that on the whole, we've been pretty crappy at coordinated biodiversity observing. There are a lot of problems. And I'm someone who works with biodiversity change data, and I can tell you that these data sets are fairly non-random. And so having bonds that are designed in a meaningful way allows us to meaningfully assess change in biodiversity across the world's oceans. Okay, so I'll take a break there briefly and see first off if there are any questions or comments either online or questions or comments from folk in the room. And I'm also going to get a drink of coffee. Yeah, Felicia.
Question. So you had said earlier, what if each network, or what if each had an experimental plot?
Yep.
What do you mean by that?
Yeah, so we'll talk about this with the manipulation, but the fundamental idea behind doing a coordinated experiment is that if every member of the network has an experiment, even if it's something simple, something that may not even necessarily stand up fully on its own as one experiment, although I think we're going to change that, so which we'll talk about in a minute. Then many experiments make a high level of replication, and if each experiment has some different set of environmental drivers, so let's say we do the same experiment everywhere around the world, everybody measures the temperature during the period of that experiment, then suddenly we have a lot of different temperatures as our covariate. And we can begin to understand something about how temperature affects the outcome of that experiment in a way that we could not previously. You can do this over smaller spatial scales, of course, but again, if your goal is to think about generality, you can only do it with that sort of level of coordination. In fact, that's a good lead-in. Let's talk about the experiment. So KEEN started off with an idea of an experiment. I'm going to talk about where we've been with that so far, and then where we're going, because the experiment has been run in about 5 to 7 sites across the world over the past 4 years. And there's some discussion about changing it. We've actually having a great conversation with Nick Shears over email talking about the tons and tons of kelp that even one manipulation has required them, and I mean literally tons, kelp that may have been moved by his students doing this project, and how changing the design could really alleviate that. And I'll talk about a possible shift in design that we'll implement here. This is not set in stone yet, so I would love feedback on this, both from the room as well as those who are participating virtually. It's a discussion that comes up every year, and so the new protocol should make it easier. But we'll talk about the old ones first because it's something that we'd still like to have as part of the network. Again, different options of different things that you can do to participate within the network. So the background of this experiment is that based on the idea that local disturbance remove kelps, will those kelps recover? Higher temperatures appear to affect reproduction in juvenile stages of kelps across taxa, and therefore recovery from local disturbance should decrease as kelps reach their thermal limit. So to put that in the context of New England, if you were to do a kelp removal experiment in, say, down East Maine, that kelp should pop back. If you were to do the same kelp recovery experiment in the Narragansett or in Long Island Sound, kelp should probably recover far more slowly. And that simply is a difference of temperature. At least that's the hypothesis. I don't actually fully have that comparison yet. So the idea really is to take each of these regions and exploit the thermal gradient in each. So each one of these regions runs from cool to warm as you move equatorward. And so if we conduct experiments along that gradient, we should be able to derive that kind of meaningful information. I laid out this sort of impact of warming of recovery hypothesis before, but of course, different regions might have different shapes to that curve. Some might be flat if there's local acclimation and nobody cares about temperature. Maybe there's sort of a linear relationship with thermal stress instead of that steep drop-off. Maybe there's variability. The reason we want to do this globally is to begin to understand is there regional variability in the shape of that relationship. So the current plan for an experiment looking at not just the response of kelps to disturbance, but also the whole kelp forest community of mobile organisms, is to conduct an experiment with many sites along a removal or along a thermal gradient with each node, each member of Keen doing one replicate. And at that replicate, have 3 control sites and 1 removal. Each removal has a common 8-meter radius, so remove all kelps within an 8-meter radius from a central point. And then sample the central 4 meters, so 4 by 4 meter area, looking at sessile species as well as mobile demersal species. So do that both before the removal, implement a removal, and then a year later, so during the next peak growing season. So broadly, take a site, find somewhere that has a depth between 5 and 12 meters, where there's kelp, where wave exposure is relatively moderate, so we aren't having variability across different wave exposure regimes, and then choose a random start point. So this is how to select a site for a Keen experiment. I know that some colleagues who I've talked to before have said, well, I'm at the southern range limit of my kelp, and kelp is relatively sparse here everywhere. So when you say where there's kelp, like I can go where there's kelp for me, but where there's kelp for me doesn't mean the same thing as kelp in, say, a much lusher bed further to the north. That's okay. We've had this discussion many times, and the fact that there's variability across a region in kelp abundance, that's all right. I mean, one thing is we'll be incorporating that as a covariate, but more importantly, that is the biology of the system. If part of the lack of kelp's ability to recover is merely it's just lower abundance for whatever reason, that is what it is. You can't sort of artificially lushen an area, flushen is a word, and then do that removal. So that is all right. But there does need to be something there to remove with, something that is representative of the ambient abundance of kelps within your particular site. So again, for this community removal experiment, the idea is to place out a 60-meter transect. And along that transect, you can locate a number of plots. So actually it's 0, 20, 40, and 60 meters on that transect. go through, sample all of the plots, and then choose one at random and remove all of the kelp in an 8-meter radius around it. That's the experiment. It's simple in terms of its design. It is, of course, a little bit harder in execution. So those members who are doing this, say, in giant kelp macrocystis-dominated systems, this is a pretty straightforward removal. Macrocystis is fairly evenly spaced, fairly broadly spaced. You take out a pair of hedge clippers, and while the sampling may take a little time, the actual removal is not that bad, and kelp floats away. I know that in other systems, for example, those that are dominated by Aclonia radiata, this is a little bit more hellish. And for those of you who have done this here in New England, where we're removing Saccharina latissima, this takes a long time because things are small and dense. So if you are interested in doing this experiment to actually look at not only the response, like this is going to give us a standardized way of looking at kelp response across the world. I still would like to include this if at all possible at sites around the world. But I understand the concerns of members who this is actually physically too much work to be done, even though it's a fairly simple design. Kelp that's small is dense and hard to remove. Particularly those of you that have the misfortune of having kelps that don't just float away on the current like we do here in New England. So I understand that. Just to sort of wrap up on this, why the design? Well, with one manipulation and 3 controls. First off, we've done this before. It takes really more 2 to 3 days with 4 divers. And 3 controls allow us to establish the range of possible change. And so we can look at an effect size of the change in the removal minus the average change in the control divided by the standard deviation in the control. And that gives us an effect size of the removal at a single site. With many effect sizes across the entire network, we can then conduct a meaningful analysis. So that's for community change. And like I said, there's an ongoing discussion about the degree to which that will continue within the network. We do have a bunch of them already, and so we're right now conducting an inventory of seeing where we have it. I think if we can do this at many places, it's going to be incredibly powerful in terms of the types of inferences we can derive. But I'll pause for a minute, see if there are any questions or comments about that removal design. And when we get to who is doing what where, we'll talk about where is doing that this year, if anywhere. Okay. This is roughly what it looks like. So we have a 12-plot layout. Each plot is 1 by 1 meter. And the reason I have these numbered is this is what we've figured out is the optimal way to sample these plots if you're rolling your quadrat around. So you sample 12 of these quads and then move on and do the removal. And there's much more on this in the handbook, which I'll reference later. Again, I just want to emphasize that standardizing conditions for this experiment is really important. There's relatively moderate wave exposure. Sites have kelp, minimal urchins, because we don't want this to be a response to urchins experiment. There are no major differences in substrate between plots. We went out and were about to conduct this somewhere and realized that one of the plots was entirely sand. And so we dropped that one and moved it somewhere else so that we had all rocky bottom. Sand makes a difference. And that the experiment is conducted between 5 to 12 meters, depending on local kelp biology. I'd emphasize that manipulation should occur at or just before natural periods of high disturbance. So we were doing it, for example, during hurricane season here in New England. The Santa Barbara Coastal LTER does something very similar to this in California, and they do it during storm season in the winter. And again, that's because we want to have the disturbance that we implement mimic disturbances that would naturally be seen by kelp forests and try and make this relevant to climate change. So if the only thing that's really different is temperature, we want to ask how does sort of natural variability or natural heavy disturbances, how does that interact with climate change to affect these forests? What has developed out of this, as I mentioned, is many people have sort of found that this size of removal, particularly for sub-canopy kelps, is too much. And so we've begun to discuss implementing something where we look at sessile species only. that size of removal for small subcanopy kelps, the only thing that is different from that is that we can start to look at mobile species. So we can look at small demersal fish, crabs, lobsters, etc. We decided not to go with a smaller removal, both because we wanted to match between giant kelp and the subcanopy kelp areas, but also because we wanted to look at those species. We wanted to see if there was change, for example, in fish usage of a habitat. If we're only interested in the responsive sessile species, so just kelps and just all of the sessile species associated with them, so other algae and invertebrates, we can do a smaller manipulation. So the idea is if we have 3 controls and 3 removals per site, so we sample a slightly smaller area, a 2 by 2 meter area, and then the radius of the removal is scaled to the size of kelps within a region, that can maybe enable us to have some more power here. It makes it easier for individual network nodes to conduct, and we still get good responses. Again, we can still look at kelp recovery, we can look at change in algae, and we can look at change in sessile invertebrates, all of which are relevant to the questions we were asking initially. Things like crabs, fish, etc., those are out the window at this size for some of the subcanopy regions, just simply because their ranges are too large. But this might be the way forward. So for example, I was chatting with Dan Smale. He's been using this design in England, and they've had because of the size of their kelps, they've been doing a 6 meter radius, and that's worked just well. Nick Shears has been doing something along this size, and it's this 3 by 3, 3 control, 3 removal design. I think it's roughly a 4 meter radius, and it minimizes the amount of work in terms of the removal while actually increasing the quality of the data. So here's sort of what that would look like. Now we have just 4 plots at the center. And I'm suggesting a 4 meter radius for New England, simply because we know that kelps will grow, they can grow up to 4 meters. So if you want to minimize edge effects, that's going to be the radius you want to go to. We could make it something slightly smaller, and I think that's something that I'd love to talk about with you guys and with other Keen 1 PIs as we go forward if we implement this. So any questions or comments about this potential alternate removal? Again, this isn't written in stone, but I wanted to present it to you guys as sort of a starting point of discussion, as well as put it on the video for other Keen members who are going to be looking at this as a potential way forward. And if you're interested, comments, either in the live chat right here, or please leave some comments on the video after it's posted. So those of you who've done this removal before, thoughts, feelings, opinions? I actually ran the calculations on this, and it's roughly half the amount of work in terms of removal than we were doing previously, even though we're upping the number of replicates. It's the same amount of work in terms of sampling, so we're actually doing the same number of quads to sample, although we're counting slightly fewer things. But it is, because of the way pi r squared works, it's half the amount of kelp removed. So it should speed things up tremendously. I don't know, thoughts? Ted, you've done this. You're up. Does this sound saner?
Yeah, I like the idea of only 4 plots counting instead of 12. I never minded the big removal that much, even though it's a lot of work.
It is a lot of work, yeah.
At Ridley Institute on Catalina, they do a lot of sargassum removal.
Uh-huh.
They use this big vacuum system. I don't know if you've ever seen that or heard of it.
Oh.
I'll show you later.
Yeah, yeah. Yeah, I'm really hoping that particularly I think that the 2 systems where this is going to be the most life-changing. I mean, one, it'll definitely change us. Again, I don't mind the removal here either, but it's a lot. It's long. It's a couple of dives to do it. Particularly in sort of Australia and New Zealand, where it's a Clonia radiata, which is heavier. And I know this will probably change things where it's laminaria hyperborea as well. I think for those places that are still working with macrocystis, keeping it at 8 meters is the way to go, because that's what you need to minimize those edge effects. That's Matt Edwards' work that says that's sort of the minimal radius that you need there. But again, things are spaced out enough that this should work. This design is also based on some work by my former master student, Jillian Dunick, who did slightly smaller radius removals and did find a lot of recovery, so that's why I wanted to increase it. But the sort of sampling design was good, and we picked up pretty good differences between plots with this sampling design.
Is it going to add on any additional like length to the overall transect?
Ah, great question. So I actually figured that out last night. And now it's a 40-meter transect instead of 60. And so, if we kind of lay it out here, if you have 0, 8, 16, 24, 32, 40, each of those has an 8-meter buffer between them. So that's 2 4-meter radiuses. So it's a shorter transect overall, so less time spent searching for your plot. Also, because it is more plots, and we do have to mark at least the center of each of these, it's going to be easier to find. One of the problems we ran into with the 60-meter transect with only 4 plots on it is sometimes it was hard to refine the transect a year later, particularly if there was heavy regrowth. More rebar in the substrate means higher probability of finding your transect line. So from that perspective, I actually feel really good about this. I think there are a lot of advantages to this design. It's just a matter of implementing it. And so we haven't talked about this yet, but if we can, we might try and do this over, say, a long weekend this fall here at UMB, something like that. Maybe this will be your last hurrah, Jeremiah, before you disappear. OK. So yeah, then choose 3 at random, do the removals. OK, any additional discussion, any additional thoughts, feelings, opinions? We've got a couple other network members online. Does any of this sound sane or insane to you? Again, we're trying to really lower the bar for participation. I mean, that's really the goal here, is to get as many people as possible to be able to feel like this is something useful. And hey, everybody needs a kelp removal in their lives, right? Especially one that now actually has replication. All right. So it's edging towards 11. It's 10:45 right now, and I promised y'all an 11 o'clock break. So for everybody online, we are going to take a break. I'm going to try and pause this Hangout. I don't even know if that's a thing. If it's not, I will come back and add a comment to this YouTube page that tells you where part 2 is. Hopefully that's not going to be the case. But right now we're going to take a break, get some coffee. Hopefully you all are caffeinating yourselves. And then we're going to talk about the surveys, data management, and collaboration, sort of the real core set of activities that everybody has been doing up until this point. So let me go here. All right, so I'm going to stop the broadcast. And like I said, if I see that I cannot restart it, I will make a note of where the new broadcast is going to be. Thanks, and we'll be back. Let's see, it's 10:45 right now, Eastern time. So let's say we will take a 15-20-minute break, and we'll be back around 11:05. So get some coffee, everybody, and then we'll come back and restart. And I will see if I can pause this.